So far, in the anchorage-dependent interactions between pancreatic cancer cells and macrophage because of tubular nanotubes, we have find out that different kind of information exchange between macrophages and pancreatic cancer cells results in different kind of characteristics acquired by the pancreatic cancer cells. And in the previous video we have discussed that some of the cells acquired these growth characteristics and some of the cells has these invasion properties. In this experiment we will understand why or what are the changes that occurs in those pancreatic cells that leads to their specific properties, such as either independent growth or these invasions characteristics. So what they did is they analyzed the transcriptome data of these separated doubly fluorescent cells with the mother pancreatic cancer cells, and then find out which of the genes that are upregulated in one cell lines and downregulated in another cell lines. So they find out that CD44 is upregulated in this SP0926 cell lines, whereas in the case of this SP1030, this CD24 is upregulated. And they validated their analysis using confocal microscopy and this flow cytometry. In the confocal microscopy, we can see that SP1030 has high CD24 populations, whereas this SP0926, even though in the protein expression, that is in the confocal microscopy, they do not see too much difference in all the three images, but in the transcriptome levels they see that in the SP0926 has high transcriptomes for the high mRNA for the CD44 in the SP0926 compared to the other cell lines, but the protein expressions are seems to be same, as there is no much significant difference, and this is the discrepancy that they have observed, but they have not discussed a reason for that. And this is the merged image. And in the flow cytometry also we can see that this pancreatic cancer cells has this 14.2% populations that are CD44 positive, and for this SP0926 they have around 3.7% populations that are CD24. These are CD24 and CD44 both positives, and in this case only 3.7% are both positives, whereas in the case of this SP1030 25.6% are positive. So indicating that this CD44 is reduced, CD24 is reduced in the case of this SP0926, whereas when it comes to CD24 it is reduced in the case of this SP1030. So all this indicates that CD44 is overexpressed in this SP1030 and CD24 mRNA levels are overexpressed in SP0926. And so in order to validate whether check whether this glycoproteins or this CD44 and CD24 plays a role towards this growth and invasion properties, they use the antibodies against the CD44 and CD24 neutralizing antibodies. Before that, the quantifications are before going into that neutralizing antibody experiment, let us go through these quantifications. So and these are also this is the combined result. B and C are the combined results. So first we will go through these images. So when they use these neutralizing antibodies against CD24, what they have find out is that there is not much difference in the tumorosphere in the SP0926, and also there is not much difference in this SP1030 because this SP0926 already has the lower percentage of this CD24 cells. That can be seen here, lower percentage of the CD24 cells. And however, when this CD24 is been knocked out, what they have find out is that cells acquired there's some increased properties of cells acquire some increased invasive properties. However, that increase is not statistically significant. And when they use this anti CD44 or neutralizing antibodies against CD44, what they find out is that it will not only reduce tumorosphere, but it will also reduce the invasion properties of this SP0926. The tumorosphere are also reduced in this SP1030 that can be seen here. And this is the combined image, and this is the quantifications where they can see that the number of cells per field in the case of these pancreatic cell lines. In the case of these pancreatic cell lines, the number of cells per field are very low. That is against the antibodies and antibodies against CD24, CD44, and the combined results are very low. However, for the case of the SP0926, the amount of cells in per field are very high for this IgG control case, and also for this alpha CD44 antibodies that are used against alpha CD44. But for this alpha CD24, for this alpha CD44, they do not see too much increase in the number of cells. So this SP0926, when the anti CD44 is used, there is no reduction in the tumorigenic effect, and the cells proliferations is also does not get affected compared to the normal case. And when they use this anti CD24, they see the slightly reduction in the invasion properties, but that reduction is not statistically significant. And this anti CD44 and the combines does not have any significant change compared to the control, indicating that CD44 does not play a role towards these invasion properties. And similar results they have found out for this SP1030. The SP1030, they see that results are in line with this control case, that is the PANC 0923. They are not statistically significant. So indicating that in this case also, in the case of SP1030, the invasion properties does not get affected by either CD24 and CD44, and this can be evident from this SP1030, where the cells almost look similar in all the cases, whereas for the case of this SP0926, the cells in first two cases are higher compared to the other two cases where anti CD44 is used. So it indicating that the tumorigenic effect is reduced when the neutralizing antibodies against CD44 is used. That can be seen here. And CD44 has a role towards this tumorigenic properties. From this figure it can be identified. And in the case of this panel B, it is the detailed descriptions of this tumorigenic properties that when they use, this is about invasion properties, and here they have discussed about tumorigenic properties. So here we can see that in the case of SP0926, the number of tumors is reduced. Number of tumors is reduced, but this reduction is not statistically significant. And also for the diameter of a tumorosphere, they can see that the results are not that the results are not statistically significant for the different cases. So this indicates that this tumorosphere are the number of the tumorosphere does not get too much affected by, in the case of this 0926, we can see that tumerosphere does not get too much affected by this anti-CD24 or anti-CD44. However, for the case of SP1030, the tumerosphere gets affected by both anti-CD24 and anti-CD44. And then about the diameter of this tumerosphere, it is also does not have too much effect, but some reduction has been observed for the CD24 and CD44. So when they knock down this CD24 using this CD24 short hairpin RNA, what they have find out is that in the case of SP1030, there is a reduction in the number of cells. The reduction in the number of cells compared to the control case, that is GFP shRNA. And this the quantification of this panel D is on the right hand side, where we can see that for the GFP shRNA, they see that number of cells per membrane are increased, whereas for this normal pancreatic cancer cells, the quantification results are as follows: that for the number of cells per membrane for the normal or mother pancreatic cancer cells, they can see that when the CD44 is used, number of cells per membrane increased. That is, CD44 has a reverse effect on invasion. And similar results they have observed when they did the experiment on the SP1030, that when they knock down CD24, they see that more number of cells per membrane, indicating that it has more high invasion properties. So this whole experiment suggests that CD44 has a role both in towards this growth and invasion properties. However, CD24 has a role towards this invasion properties, and that occurs during anchorage-independent cell transfer between macrophages and pancreatic cancer cell lines. So I hope you understood how these experiments goes through and what are these CD24 and CD44 functions in the anchorage-dependent cell transfers between macrophages and tumor. If you need to understand more about this experiment in detail, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.